And the next presentation is by Lotte Wheaton from Holland. The 2-tango antibody-dependent cell-mediated cytotoxicity enhances superiority of allogeneic active NK cells against multiple myeloma.
Thank you for the introduction and good morning to everyone. First and foremost, I would like to thank the scientific committee for having selected our work for this prestigious session. Allogeneic hematopoietic stem cell transplantation is the best therapeutic option for many patients suffering from hematological malignancies. And this is largely due to the graft versus leukemia effect. Still, post-transplantation relapses remain an open issue. In the context of HLA haploidentical transplantation for acute myeloid leukemia, in 2009, Luca Vago and Katarina Fleischauer described a novel and frequent mechanism of relapse, the HLA loss relapse. Prompted from the intense immune pressure mediated by alloreactive donor T cells, leukemic cells can undergo de novo genomic loss of the mismatched HLA haplotype through a mechanism of acquired uniparental disomy. This mechanism completely abrogates leukemia recognition by donor T cells, allowing the malignant blast to evade the immune system and ultimately foster leukemia relapses. As some of you already know, landmark studies from Perugia Group have demonstrated that in the context of T-cell depleted haploidentical transplantation, NK cell alloreactivity have an important role to prevent leukemia relapses. In the particular context of HLA loss relapses, this can become even more relevant, since NK cells that at the time of transplantation were inhibited by the presence of specific HLAs on the surface of the target cells can become alloreactive when the mismatched HLA haplotype is lost. Thus, the aim of this study was to investigate the NK cell dynamics in this very peculiar context and understand why NK cells alone are not able to prevent or control the occurrence of HLA loss relapses. We analyzed the complete series of HLA loss relapses documented in our center, now counting a total of 30 cases. Most of them are patients suffering from myeloid malignancies and undergoes haploidentical transplantation. But as you can see, there is also a case of ALL and 2 cases of HLA loss relapses after partially incompatible unrelated donor transplant. Finally, I would like to point you out another peculiarity of this type of relapse. They occur very late after transplant at a median of 300 days. First of all, by using the Perugia algorithm, we checked our series for potential NK alloreactivity that was predicted in the 40% of patients at the time of transplantation and gain at the time of HLA haplotype loss in additional 23%, accounting for a total 63%. Next, we address whether the lack of NK cell-mediated control of HLA loss relapses was due to the lack of activatory kills in their donors. For this purpose, we performed key genotyping of all tilted donors for HLA loss patients, documented that actually the frequency of KIR-B haplotype was even higher than in general population, suggesting that the defective immunosurveillance mediated by NK cells cannot be explained by an unfavorable immunogenetic assets of donor. Thus, we developed a very comprehensive immunophenotypic panel, including 27 surface markers, and employed it to study 4 experimental groups. Patients at the moment of HLA loss relapses, patients at the moment of non-HLA loss or classical relapses, patients in remission, and healthy control. Please note that both classical relapses and patient in remission have been selected to be studied very late after transplant, just to be comparable with the group of HLA loss relapses. Results were also analyzed and validated with BH-SNE, a bioinformatic tool that allowed to condense very complex multiparametric data in a simple bidimensional maps. The most interesting results were obtained for the markers highlighted in red. According to the Perugia model, alloreactive NK cells should be positive for a KIR missing its ligand on target cells and negative for all the other inhibitory receptors for which the ligand are present. Using these gating strategies, we studied the frequency of single KIR positive NK cells within our group, and in particular of those potentially alloreactive shown in red. As you can see, potentially alloreactive single KIR positive NK cells is present in all experimental groups, although the frequencies are very variable. And in particular, they are present also in the peripheral blood of patients with HLA loss relapses. So we have to look as well. And interesting, we found that Siglec-9, a receptor belonging to the lectin family, recently showed to be important in NK cell-mediated immune surveillance of solid tumor, is slightly quite almost absent on NK cells of transplanted patients. Also, when we studied those patients at 3 years after transplant, when the immune recovery is supposed to be complete. This result is also evident on this Bisney map. In fact, as you can see, the cluster of NK cells that highly express Siglec-9 is very evident on the map of healthy control and almost completely absent on the maps of transplanted patients. We obtain another very interesting result regarding TIM3, an inhibitory receptor particularly studied to be an exhaustion marker on T cells, and the upregulation of which has been linked to NK cell functional inhibition. As you can see on both this dot plot and Bisney map, the TIM3 is highly upregulated on NK cells of HLA loss relapses compared to classical relapses, suggesting that the lack of initially alloreactive NK cells can become exhausted over time. In line with this hypothesis, we collect very preliminary results showing that NK cells of HLA loss patients display lower frequency of both TBET and EOMES, two transcription factors shown to be fundamental not only for the immune cell fate determination, but also be tightly linked to functional exhaustion. In conclusion, I show you that even though NK cells of HLA loss patients are alloreactive at the time of transplantation, this was not enough to prevent clinical relapses. This defective immunosurveillance cannot be due to the lack of activatory kill genes or single kill positive NK cells, but might be related to functional exhaustion. Our results suggest that the NK cell repertoire circulating in HLA loss relapses are largely defective. As a solution, we propose that therapeutic protocols employing freshly isolated mature donor NK cells should be tested for the prevention and treatment of HLA loss relapses. To conclude, I would like to thank all the collaborators, including all the hematology unit headed by Fabio Ciceri and the group of Katharina Fleischauer in Essen. And last, I would like to thank all the members of my groups, in particular Giacomo Oliveira and my boss Luca Vago. And thank you all for your attention.
Valentina, thank you very much, and I deeply apologize for introducing you as the next speaker. But you sorted that out.
Okay, no problem. Thank you.
Are there any questions or comments? If not, could I ask you, do you have any functional